Okay, so let's continue with the first week. Now we're going to talk, just now we already discussed about the gametogenesis, and now we're going to look at the ovulation process and fertilization. Okay, so what is fertilization? Fertilization is the process whereby the sperm and ovum are fused together. Okay, so that is what is fertilization is. And usually female produce only one secondary oocyte per month. I already mentioned it in a previous video that unlike males who are producing the millions of the sperm throughout their life, starting from the puberty, okay, but the female only produce one in each month, okay, in one each month. And what happened is that when the ovulation takes place, the secondary oocyte need to travel along they've been ovulated from the ovary, isn't it? From the ovary, they need to enter into the uterine tube, the fallopian tubes. Okay, so when they enter the fallopian tubes, they will travel towards the uterus, okay, where the implantation will take place. So this will takes around 3 days, and the lifetime for the secondary oocyte is not that long. It's about 24 hours. As a result of that, the sperm need to find the needs to search or find the ovum, the ovulated secondary oocyte. So that's why, coming back and giving you the clue, the question that I gave you during the second part of the video. So that's why the shape of the sperm is very slender. Then because the function is to find and swim, get very close to the secondary oocytes. Okay. And if the fertilization, so the fertilization usually occurs in the fallopian tubes, and if you look at the anatomy of the fallopian tubes, they have different segments, and the ampulla is the most common area where the fertilization can take place. Okay, so if the fertilization take place, so what happened is that the secondary oocyte, the fertilized oocyte, needs to go towards the will move towards the uterus for the implementation. So the sperms, if you look at here, I don't know who counted, but I just believe with the reference that I read on, male produce 300 million sperm per ejaculation. This is a huge number, but out of this number, only 200 or 2000 or 3000 spermatozoa that manage to get to very close to the vicinity of the ovulated secondary oocytes. Okay, and out of this number, only one lucky spermatozoa that manage who first touch the zona pellucida of the secondary oocyte will be the one that fertilize the ovum. Okay, so there are certain conditions that the sperm need to undergo before they can manage to touch the finish line. Okay, so this process we call it capacitation. Capacitation is a process whereby the sperm undergo biochemical and physiological changes on the surface, on the membrane of the sperm. Yeah? And what happened is that they remove the glycoprotein so that the receptors are available. At the same time, they also undergo a chromosome change, whereby the chromosome become very permeable, and so that it can release the lytic enzyme that can penetrate the wall of the secondary oocytes. Okay, so what happens? So perhaps I can show you the picture here. Okay, so you can see this is the secondary oocyte, and as I said, during the oogenesis, they will produce one huge secondary oocytes with very small first polar body. I don't want to give you the answer why first polar body is very crucial, but perhaps I can give you the clue that is related to the chromosome number. Okay, so this oocyte is actually covered by a zona pellucida. There is a one zona pellucida. So as I said to you, the first sperm that managed to touch the zona pellucida will be the one that will finally fertilize the secondary oocytes. Okay, surround the zona pellucida, you can see there are a layer, one or two layers of a cell. We call it corona radiata. Okay, so corona radiata. So this you can see only few that manage to get the vicinity of the secondary oocyte, but this is the one that the first that penetrate the oocytes. Okay. So let's have a look at the phases of fertilization. What happens is that the sperm needs to make a tunnel, okay, passing through. They have to make a way around the corona radiata here. So there are a lot of cells here. So in order for them to penetrate this layer, they need to produce an enzyme, and this enzyme we call it hyaluronidase. Okay, this is the hyaluronidase. Then by using their tails, they force themselves towards the secondary oocytes. Yeah, and then the next step is that penetration of the zona pellucida. Okay, so this is the process whereby another types of enzyme, proteolytic enzyme, we'll call it acrosine. This is the enzyme that will make a tunnel, okay, to the zona pellucida. And what happened is that, okay, for example, this one, yeah, this managed to penetrate the corona radiata, and then when they reach the zona pellucida, they will produce another enzyme, the acrosine, and the acrosine, by releasing this enzyme, they can make tunnel so that they can make a way into the secondary oocyte. What happened next is that the first sperm that touch this zona pellucida will leads to a zona reaction. Okay, what's zona reaction? Zona reaction is the condition whereby it's a process whereby the rest of the zona pellucida here become hardened. So meaning, even though this particular sperm also touch the zona pellucida, but they cannot enter, even though they produce the acrosine, they cannot enter because already this area is already become hardened. Okay, very hard, so they cannot penetrate. Only this one that can allow be enter into the secondary oocyte. Okay, so that's the zona reaction mean. Then what's the next? What happens next is that the plasma membrane of oocyte and sperm will merge together, and as I said, the oocytes are the kept at the metaphase 2 during the meiosis 2. So once the sperm manage to fertilize the secondary oocytes, the oocyte will undergo complete their meiosis 2, and then they will produce the form female pronucleus. Yeah? And male also have the pronucleus, and they form the oocyte. So what is oocyte? Oocyte basically oocytes that have two nucleus, yeah, pronucleus. One is from the male, female, and this is from the male, the one sperm that enter into it. Okay, so from here now it's become zygote. Okay, they are fertilized, they already complete the meiosis 2, they become zygote. Okay, and then from the zygote, the next step will be the formation of the zygote will undergo the cleavage process. Okay, cleavage process is basically like mitosis. They undergo multiple number. Yeah, they undergo a series of division to form multiple cells. Yeah. Okay, so perhaps I can explain about the cleavage by showing you this picture. Okay, you can see this is the ovary. They produce the ovum. This is the ovulated secondary oocyte. They will travel into the uterine tubes, and then at the same time, the sperm, when the intercourse takes place, the ejaculation, the sperm will be travel into the uterus and then passing through the uterine tubes and to search for the ovulated secondary oocyte. So the fertilization takes place here, and then the cleavage will be the next step. Okay, so from one cells, zygote, they divide into 2 cell stage, 4 cell stage, and 8 stages. So this is we call it cleavage process. At one point, you can see that they become morula when the cells is about more than 12 to 16 cells. We call it morula. So morula is a very a ball of cells that can have a lot of cells inside. Yeah? And these cells, later on, they will undergo the formation of the blastocyst. So how to differentiate between morula and blastocyst is the moment when the fluids come into this ball. Okay, when the fluid comes into this ball of cells, we can start to see space. So this is the early stage of the blastocyst. So how to differentiate morula and blastocyst is by having fluid inside the ball. Okay, fluid stick into the balls. Okay, so if you cut this ball, you can start to see the cells also undergo differentiation. They differentiate into two. Okay, this is, we call it inner cell mass, or also known as the embryoblast. So embryoblast is the cells that will develop into the embryo. So the names is actually reflects the what's the fates of the tissue. Okay, and the blue color here is, we call it trophoblast. So from these cells, a pack of cells, they undergo differentiation into these embryoblasts and the trophoblast. Trophoblast will be the one that form into the placenta and extraembryonic membrane. Yeah, the embryoblast here is the one that will develop into the embryo. Okay, so as they go further down, okay, as they undergo the cleavage and the formation of the blastocyst, these cells actually they travels towards the uterus, because the end product is that the blastocyst need to be implanted on the wall of the the wall of the endometrium. So you can see there are another stages at the late stage of the blastocyst. You can see the embryoblast also, they split into two. We'll talk that shortly. And then you can see the size become bigger, and you can see that this is the zona pellucida around it also been dislodged. Okay, they've been removed in order for the late blastocyst to enter or implanted into the wall of the uterus. Okay. Okay, so this is another picture. You can see that this is the blastocyst. It looks like a ball like morula, but morula is completely cells inside. But blastocyst, when you cut, you can see this kind of pictures. Okay, you can see this is the trophoblast, the one that will develop into the extraembryonic membrane, placenta, and so on. And this, the blue color here, is the embryoblast. It's the one that will develop into the embryo. Okay, so next, this is the late stage of the early stage of the blastocyst. Okay, as they go along on the uterus, you can see they try to implant themselves on the wall of this is the wall of the endometrium. So you can see the trophoblasts undergo differentiation. Okay, this time they form into two different types of cells. The one is brownish color here, we call it syncytiotrophoblasts, and the blue color here we call it cytotrophoblast. So the difference between these two? The cytotrophoblast is like columnar cells, simple columnar cells. They have very nice cell membrane in between cell wall between the cells. But syncytiotrophoblasts, they are just a multiple number of cells, but there's no demarcation, there's no cell wall in between the cells. Okay, they do have different role in this regards. Yeah? The syncytiotrophoblasts is the one that produce the enzyme that can engulf, excavate the wall of the endometrium, so that they can bring the whole embryo blastocyst into the endometrium wall. Okay, so this is the cells that are responsible to produce enzyme that can engulf, make a tunnel, okay, that can excavate the wall of the endometrium, so that the whole blastocyst can implant it into the endometrium. Okay, so what's the function of the cytotrophoblast? Cytotrophoblast is the one that produces a hormone that helps to maintain the corpus luteum in order to maintain the wall of the endometrium here. So if you still remember the cycle, the ovarian cycle, there are hormones that helps to maintain. I already mentioned you in the first part of the video. The hormone is very important to maintain the wall of the endometrium, to maintain the thickness of the wall of the endometrium, so that it's ready to receive the implanted embryo, the blastocyst. So I stop there. Continue in the next videos. Okay, thank you.